Hello children, welcome back. Today we are going to discuss the second chapter, sexual reproduction in flowering plants. Are we not lucky to visualize these flowers, these beautiful flowers, the fragrance they produce and the scents they give? We swoon over them, right? And also they produce many vibrant colors which attract us. So have you ever thought why these features are existing in flowers? Why flowers do have these features? It's for us to visualize and have visual treat and having their fragrance and scents? Definitely no, right? We may think like that, but plants do have a life. These flowers do have a life. They have to reproduce sexually. Yes. So these flowers are having these features because they help them in sexual reproduction. So that is what we are going to discuss today. We are going to discuss the morphology, structures and events of fertilization and sexual reproduction in flowering plants. Come on, let us go to our class. So we are going to talk about a flower, androecium, microsporogenesis and pollen grains. If you would recall, so flower is a fascinating organ, right? So what is cultivating of flowers generally? Only for cultivating purposes and not for economic purposes. What is it called as? It is called as floriculture, right? To increase the breed like flowering varieties and all those things, only for gardening purposes and no commercial purposes, floriculture is done. What do floriculturists do? They just try to increase the varieties and breeds of flowers. So we can just go for a short recall from your younger classes. What have you learnt about flowers? So yes, the flower contains 4 major parts which you know: petal, sepal, stamen and pistil, right? So here you would have learned from younger classes that petals and sepals, we are just going to name them as calyx and corolla. The sepals are calyx and the petals are corolla. Okay. So the stamen is termed as androecium and the pistil or carpel is called as gynoecium. You can see there that the stigma is having, the pistil is having 3 parts: stigma, style and the ovary. Similarly, a stamen has 2 parts: anther and the filament. Okay. So we are now going to discuss about the pre-fertilization structure and events. So what should happen before fertilization? If you could remember from the last chapter, first a gamete should be formed, then only it can fuse to form a zygote. Similarly, in plants we do have gametes. We have male gametes from male, that is the stamen, and we have ovum from the pistil. So where and how these are formed, we are just going to discuss about that in the beginning. Then we are going to discuss about the fertilization. So in the flower, the male and female reproductive structure, the androecium and gynoecium, differentiate and develop. So we would recollect that androecium consists of stamens, right? So we are just going to discuss about stamen, microsporangium and pollen grain first, followed by our gynoecium. So you can recall this. So this structure you would have studied from your smaller classes, right? So this is an anther. So how anther will be? Anther will always have a bilobed structure, right? You would have learnt this. Then it has a long stalk called as filament. So these are separated. So each lobe contains 2 lobules in them and are separated by a long line. You can see this, and they also contain a filament or stalk. So these filament will vary in length in different species. If you can collect different species like flowers, stamens from different flowers, you can see that the length of the filament, the place of their attachment will show different variations. So yes, a stamen consists of a long slender stalk called as the filament and terminal generally bilobed structure called anther. So the proximal end of the filament is attached to the thalamus or the petal of the flower. So as I said, the number and length of stamens are variable in different species. So a typical angiosperm anther is bilobed with having 2 theca. So I said lobules, so they are called as theca and not lobules. So each lobe will contain 2 theca and they are called as dithecous. Since they are having 2 theca, they are called as dithecous. I said a longitudinal line will be running in between them to separate these theca. So they are called as longitudinal groove. So an anther typically has a tetragonal structure consisting of four microsporangia that are located at the corners, like 2 in each lobe will be present. So these microsporangia will further develop to form pollen grains, and after dehiscence of anther these will be released out so that the fertilization can happen. So if you can see, these are the layers of a microsporangia. So these microsporangia have something in middle which is called as connective. So we can just go into the structure of microsporangia. So these will have some sterile cells in between. So generally they just appear like circular when you see from outside, but they are not circular, they will be tetragonal, and you know that, right? So their structure will be something like this. I am representing it for one side and you have to take it for the entire side. So the first layer or the outermost layer will be epidermis, which is followed by endothecium. So you would have seen something called connective which is present in middle, right? So some cells, some sterile cells will be present in middle, and these cells are called connective. So following the epidermis you will have endothecium. So endothecium lining will be followed by the epidermis. Okay, so next after this you will have middle layers. So middle layers will not be single layer, it will be around 3 to 4 layers of cells will be present in the middle layer. You can just imagine this will be present for the entire anther, will be present in the entire surface of the anther. So these are the middle layer which I am drawing now, and these points here and here are called as the points of dehiscence. So after that what will be present? You will have a layer which is called as tapetum. So these tapetum are generally conical. They contain a dense cytoplasm and they have a large number of nucleus. Why do they have a large number of nucleus? Because they undergo endomitosis. Their nuclear division will not be followed by cytokinesis. So the work of this middle layer is basically to provide nutrition for the developing microsporangium. Okay, so when the anther is young, there are some tissues present here which are called as sporogenous tissue. So let us recall from the first. So the outer layer will be epidermis, the middle layer will be endothecium, sorry, not the middle layer, the next layer will be endothecium. So in between you have middle layers. So middle layers will be 3 to 4 layers thick. So after that you have tapetum. Tapetum is always multinucleated. They have dense cytoplasm and large number of nucleus. Why they have large number of nucleus? Because they undergo endomitosis, and these are called as sporogenous tissue when the anther is young. So once when the anther is starts to mature, it is called as pollen mother cells. So these cells are capable of giving rise to pollen grains. So after once they are matured, they are called as pollen mother cells. So all the 4 sites only sporogenous tissue will be there. Everything will develop into pollen mother cells. So for understanding, I am drawing it like this. They will develop into pollen mother cells, or you can even write it as PMC. These pollen mother cells will undergo meiosis to produce 4 pollen tissues. They are microsporous tissues, and they are generally called as microspore tetrads because they will be like 4 in number. So here I am drawing 3 surfaces, right? So we have various arrangements. So these 4 tissues or 4 pollen grains, microspore tetrads, will be arranged in various arrangement which we will be discussing later. These pollen tetrads mature and then they lose water, that is they dehydrate and form 4 pollen grains. Clear? So they form 4 pollen grains. So this is a pollen tetrad or microspore tetrad. Tetrad is nothing but because they are 4 in number, and these are the pollen grains. Once the anther matures, what happened? These both are the point of dehiscence, right? So they burst out and then they will be released so that they can go and fertilize. You can see here, these are the point of dehiscence. From here the pollen grains are being released. So yes, structure of a microsporangium. A typical microsporangium, as I said, will appear only circular in outline, but it is surrounded by 4 wall layers: epidermis, endothecium, middle layers and the tapetum. The outer 3 walls perform the function of protection and help in dehiscence of anther to release the pollen. As I said, the innermost layer or the tapetum, what it will do? It will nourish the developing pollen grains. Cells of tapetum possess dense cytoplasm and they generally have more than one nucleus. So when the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue. These things I have mentioned. The sporogenous tissue will occupy the center. So occupies the center of each microsporangium. So what is microsporogenesis? As the anther develops, the cells of the sporogenous tissue undergo meiotic division. So that is reduction division to form what? Microspore tetrads, which I have already told you. So these microspore tetrads, what they will do? They will dehydrate and form the pollen grains, right? So each cells, as these sporogenous tissue, when they mature to become a pollen tetrahyd, they are called as pollen mother cells, or called as, you can mention it as PMC. The process of formation of microspores from a pollen mother cells through meiosis is called as microsporogenesis. The microspores, as they are formed, are arranged in a cluster of 4 cells called as the microspore tetrahyd. So these things only I have told that I will be mentioning in what arrangement they are present. So here 3 tetrads out of 4, 3 will be present in the front and 1 will be present at the back side. So else 4 will be arranged like that. So this structure is called as tetrahedral. This structure is important because many pollen grains or microspore tetrads will be generally in this form, that is tetrahedral condition. So the second condition is isobilateral condition and the third one is called as the decussate. 3 will be on one side and again 1 is on the back side. Then this is a T-shaped microspore tetrad. You can see 2 are arranged on the top and the other 2 are in the bottom. Then all the 4 will be arranged in a linear fashion and that is called as the linear arrangement. So as I said, the anthers mature, dehydrate and the microspore dissociate from each other, forming the pollen grains. So inside each microsporangium, what will be present? Several thousands of pollen grain will be present, which are released during the dehiscence of anther. So now we are going to discuss about the structure of a pollen grain. The pollen grain generally has a outer hard covering which is called as exine. So you should remember this is an hard covering because it contains a substance called as sporopollenin. So sporopollenin is a substance which cannot be degraded by any enzymes which are discovered so far, and also it is highly resistive to temperature and acids, strong acids and alkalis. You have to remember, since pollen grain is having these sporopollenin, it can be stored as a fossil even. Okay, so this is the exine layer which is hard and presented outside, which is having sporopollenin, and there is a small groove like thing in this which is called as the germ pore. This germ pore is going to develop as the pollen tube when it is going to land on stigma. Okay, so the inner layer is generally called as intine. So there will be a nucleus present here which is called as a vegetative nucleus. So this is the nucleus, so I am just shading it like this. So this nucleus is generally called as the vegetative nucleus. So apart from that, there will be a small cell present in this pollen grain which is called as the generative cell. An entire small cell will be present. Generally these are like 2 cells. So this cell is called as the vegetative cell containing vegetative nucleus, and this cell is small and it will float in the cytoplasm. This is the generative cell. What cell is this? It is generative cell. It floats in the cytoplasm. So basically this cell again contains a nucleus which is called as generative nucleus. So again it contains generative nucleus. We saw the structure of pollen grain. Again some vacuoles will be present. So in many plants what will happen, here two nucleus are present. So I am shading the nucleus alone. Here two nucleus are present, right? Pollen grains in some plants will be shed as in two nuclei stage, okay? In two cell stage itself they will be shed. Whereas in some plants what happen, this nucleus, this generative nucleus will divide to form two male gametes, okay? So these divide to form two male gametes. So sometimes after the release they will divide and they will be forming two nucleus here. So in some plants before release there will be a 3 nuclei form, but in some plants only after their release there will be a 3 nuclei form. You can see these are microsporogenesis and microgametogenesis. So microgametogenesis is nothing but the formation of the released microspore is being forming into a tricellular pollen which is having 3 nucleus there. A vegetative nucleus and one generative nucleus. What happens to the generative nucleus here? The generative nucleus will divide into two male gametes. You can see the structure of a pollen grain here. You can see the germ pore, generative cell and generative nucleus. So this is the vegetative nucleus. We are having vacuoles even. So yes, pollen grains are like 25 to 50 micrometers in diameter. So it has a prominent two-layered wall. As I said, the outer layer exine is very hard and it contains a compound called sporopollenin. As I said, it is the hardest substance and no enzyme is still found in the earth to degrade that. Because of this character, the pollen grain is stored as a fossil fuel. Okay, so then it can also withstand high temperatures and strong acids and alkalis. So pollen grain hexane has a prominent aperture. So as I said, a prominent aperture will be present which is called as germ pore. In germ pore, sporopollenin will be absent. Okay, so as I said, pollen grains are well preserved as fossils because of the presence of sporopollenin. Then one question, why do you think that hexane should be very hard like this in nature? Why? Because during pollination, these pollen grains will be traveled and carried to many places which are having high extremities. So to withstand them, to protect the pollen grain, the hexane, to protect the male nucleus and the developing nucleus or the developing gamete, the pollen grain is very hard. So now the inner wall of the pollen grain is called as the intane and it is made up of cellulose and pectin. So the cytoplasm of pollen grain is surrounded by plasma membrane. So the pollen grain is mature. It contains two cells, as I said, one is vegetative and the other one is generative. The vegetative is bigger and contains more amount of nutrition. When you take generative cell, it is very small and it floats in the dense cytoplasm. So it is generally spindle shaped with the cytoplasm and nucleus. In over 60% of angiosperms, pollen grains are shed at this two-celled stage. In the remaining species, the generative cells divides mitotically. So you should see this. The generative cells will divide in which way? They divide mitotically to give rise to two male gametes before pollen grains are shed in a three-celled stage. So finally, is pollen grain injurious to health? Do you think that they will have any adverse effects? Yes, they have. These pollen grains can cause asthma and bronchitis. Okay, they can lead to asthma and bronchitis and they are also reported to cause severe skin allergies. So it may be like they have told in India that Parthenium or carrot grass. So carrot grass came along with imported wheat. So after that it became a normal plant in India. So ubiquitous. It can be seen in everywhere and easily. So this plant is said to cause more amount of pollen allergy. So my next question will be, how viable are pollen grains? So in many places you can see sperms of many species like human beings, cows are being stored for artificial insemination. Can pollen grains be stored like that? Yes, of course they can be stored at a temperature of minus 196 degrees Celsius in liquid nitrogen and used as pollen banks for breeding programs. So the period of which pollen grains remain viable is highly variable to some extent. So some will live for many long years, whereas some live only for a short period. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release. They only live for 30 minutes, and in some members they remain even for months. Okay, so as I said, it is possible to store pollen grains of a large number of species for years in liquid nitrogen in 196 degrees Celsius. It is 196 degrees Celsius. Yes, such pollens can be used as pollen banks similar to seed banks in crop breeding programs. So, do you think these have any other advantages? Of course, yes, they are also taken as supplements in many Western countries. So, these pollen grains are taken as food supplements. Since they have many nutrients, they are taken as food supplements in many countries. So, as I said, pollen grains are rich in nutrients. So, it is now a fashion in recent years that pollen tablets are used as food supplement. So, this is majorly done in Western country. So, particularly for what? It is also found to increase the performance of athletes and race horses. When you give this tablet regularly for athletes and race horses, it has said to increase their performance. So, these are some pollen tablets which are taken as supplements. Come on, let's have a small recap. We have studied about the male part of the flower. We also saw what are pollen grains, how are they formed, what is microsporogenesis. We also discussed about what are the advantages and disadvantages of pollen grains. We also saw that pollen grains contain an important compound called sporopollenin, and pollen grains can be stored for many years in pollen banks. Okay, in next class we will discuss about the female part of the flower, which is gynoecium, and we will also see how the egg is formed, what is fertilization, and before that we will also discuss about an important event called pollination. Let's see it in next class. Thank you, children.
Please contact us, your buddy, on 98408 44987.